Gene mutations are very important in cancer because they essentially cause cancer to happen. So what gene mutations are, they are changes in the genetic code in the cells that give our cells instructions on how to behave. These instructions tell the cell when to grow, when to rest, when to die. So when gene mutations happen, they result in the changes in the code that changes the protein that the cells make. So these new proteins, abnormal proteins that result from gene mutations, can tell the cell to grow too much, not to die, and to metastasize to other places in the body. So in this way, these gene mutations actually are the hallmark of cancer cells. They can be categorized, in various ways. But to understand that, I think one of the things that is another hallmark of a cancer cell is that it grows uncontrollably. It divides when it's not supposed to, and it also does not die when it's supposed to. Our healthy cells are very well controlled. They only grow when they need to grow, and they need to replace, dead and dying cells. And they also die when they're supposed to, so that they don't live too long and cause problems in terms of accumulation of cells in the wrong places. However, gene mutations such as tumor suppressor genes, which are proteins that hold back the growth of cells, they act as brakes on the proliferation of cells. When you mutate these genes, they take the brake off the cell growth, and the cells grow uncontrollably. The other type of genes that drive cancer would be a type of gene called the oncogenic gene. These include genes that cause proliferation of the cell by driving cell growth, or genes that prevent cell death. So you can imagine that if you cause a cell to proliferate, it can also increase in number. But if you also prevent the cell from dying, you can also cause accumulation of the cell, increasing the cell number. One of the examples of an oncogenic gene is the KRAS G12C mutation. The KRAS gene is very important in cell proliferation, and when you mutate this gene, you will cause uncontrolled growth of the cell. Other types of gene mutations that can occur that lead to cancer include mutations in anti-apoptotic genes. Apoptosis is a natural process of cell death and happens to cells to control its numbers. When you mutate anti-apoptotic genes, you cause the cells not to die when they're supposed to and cause accumulation of cells. This is also a hallmark of cancer. The last category of gene mutations that are important in cancer would be mutations in DNA repair genes. DNA repair genes are responsible for accurate reproduction of our genetic code when cells divide. So when you get mutations in these DNA repair genes, you have more mistakes in DNA replication, and this results in more mutations. And these mutations can result in mutations in tumor suppressor genes or oncogenes, which then lead to cancer. A common example of this would be the BRCA gene, which can increase the risk of breast cancer, ovarian cancer, and pancreatic cancer. As we stated before, the KRAS gene is a very important gene in controlling proliferation of cells in the body. When you have mutations in the KRAS gene, such as the KRAS G12C mutation, you end up with increased proliferation of cells, increased survival of cells, and decreased differentiation of cells. This leads to cancer. There are different types of KRAS mutations, and the KRAS G12C gene accounts for about 44% of all KRAS mutations. At ICON, we are dedicated to providing our patients access to clinical trials and invest in clinical research across our cancer centers. We are currently performing 2 clinical trials targeting the KRAS G12C gene for both non-small cell lung cancer and colorectal cancer. They are open and currently accruing patients at our centers. For more information, please visit our website